In our final screencast on chapter 13, we're going to look at how the process of meiosis introduces genetic variation into a population. And this is extremely important for maintaining species and for evolution. Okay. All genetic variation produced in sexual life cycle contributes to evolution, and pretty much all of it starts with a mutation. So mutation is a change in an organism's DNA, and this is the original source of genetic diversity. When you have a mutation, it creates a different version of a gene called an allele. And reshuffling of alleles, creating different combinations of alleles during sexual reproduction, produces genetic variation. So we're going to look at a couple of ways that we do kind of this step, this reshuffling of alleles. Okay. A lot of genetic variation comes from how the chromosomes move, or the behavior of the chromosomes during meiosis and during fertilization. And that is, again, almost all of the variation that arises in each generation. And there are three main mechanisms that contribute to genetic variation. One is independent assortment, 2, crossing over, and 3, random fertilization. So let's look at independent assortment first. Okay, so basically independent assortment refers to how the homologous pairs of chromosomes orient themselves during metaphase 1. So how they line up with which chromosome kind of is facing which pole is completely random. And this we call independent assortment. So each pair of chromosomes sorts the maternal and paternal homologs into the daughter cells independently of all other pairs. So the number of combinations possible when the chromosomes assort independently into gametes is 2 to the n power, where n is the haploid number. So for humans, n equals 23, which means there are more than 8 million combinations of chromosomes that can result in forming your 4 gametes. There are 8 million combinations that are possible. And again, here we can look at this is a cell that only has a haploid number of 2, n equals 2, because there are 2 chromosomes. So there are only going to be 4 possibilities, because we get 2 squared equals 4 possibilities. So if we start off scenario 1, all the paternal chromosomes are on one side, all the maternal chromosomes are on the other side. So we separate the homologous pairs in meiosis 1. Okay, and then we separate the sister chromatids in meiosis 2. And so again, how they line up during metaphase 1 is completely independent of each other. And that gives us these 2 combinations. Okay. Another possibility is that the chromosomes can arrange themselves in a different way, where you have 1 paternal and 1 maternal on this side, and 1 maternal and 1 paternal on that side, so that during meiosis 1, when we separate the homologs, will result in these different combinations of chromosomes. Separate the sister chromatids in meiosis 2, and that leads to 2 different combinations. So a total of 4 combinations. And imagine doing this with 23 pairs of chromosomes. It can get a little dicey because you have to come up with 8 million different combinations. Okay, so that is one way of creating genetic diversity. Way number 2 is crossing over. And this happens during prophase 1 of metaphase 1. And the process of crossing over produces recombinant chromosomes. So recombining, making new combinations of alleles. Okay, and recombinant chromosomes combine the DNA inherited from each parent. So you don't have anymore a chromosome from mom and a chromosome from dad. You have your chromosomes that you will pass on to your offspring, and they are completely novel. So crossing over begins very early in prophase 1 as homologous pairs pair up. This is called, again, synapsis. And then once they're paired up, they cross over. Some homologous portions of non-sister chromatids trade places. They swap little pieces of each other. And crossing over contributes to genetic variation by combining DNA from two parents into a single chromosome. So how this looks, we have here non-sister, so we're in prophase 1. Non-sister chromatids held together during synapsis. Then the points where the material is going to exchange are called chiasma. And basically they break and switch places. And so now we have recombinant chromosomes with the sister chromatids are now no longer identical. Okay, so the homologous pairs separate during anaphase 1. The sister chromatids separate during anaphase 2. And we're left with 4 combinations, but 2 entirely new chromosomes. Okay. The third way of generating genetic diversity is random fertilization. Okay. So random fertilization adds to genetic diversity because any sperm with any genetic combination can fuse with any egg with its own genetic combination. Okay, so each gamete, you remember, has from independent assortment has about 8.4 million possible chromosome combinations. Then that's from independent assortment. If you multiply the 8.4 million by 8.4 million, you get about 70 trillion diploid combinations for a zygote. And then if you add crossing over on top of that, which is going to create all new chromosomes in the mix, the possibilities are literally infinite. Okay? And because of that, every zygote has a unique genetic identity. When you say you're one of a kind, it literally means that, unless you're a twin. So here's our little evolutionary piece. Evolutionary significance of genetic variation, super important. And when we get to studying evolution, we'll talk about this again and again and again. So natural selection results in the accumulation of genetic variations that are favored by the environment. And that's why we have to have so many possible genetic variations, because we don't know what the environment is going to throw at us. So we can't change our DNA if the environment changes. We have to hope that we have something in our DNA that will be useful if the environment changes. Okay? And this is all from sexual reproduction. Sexual reproduction contributes to genetic variation. And again, it all starts with mutations. And that's it for Chapter 13.